Hi guys, welcome to the YouTube channel. My name is Sarmad Haider. I am a biomedical sciences major at Marquette University, and I wanted to start this channel to just work through some biochemistry problems because I'm in my gap year and I'm bored as heck, and I was like, I want to keep up with the sciences. I love biochemistry, I love biology, I love chemistry, and I just want to continue learning and teaching. And teaching is something that I love doing. I taught a lot during my office hours for my professors. I just love conveying this information because I find it super interesting, and I want you guys to find it super interesting as well. So we're on this journey together. I have a webcam or a camera here so you can see me while I solve it on the iPad. And basically the way I'm going to be making these videos is I'm going to have a question and then a what we need to know. And this question is from a textbook from the MCAT AAMC textbook that they recommended for biochemistry. This is chapter 2, so we're talking about amino acids. And I just wanted to pick up a question that I thought conveyed some really important topics that you have to know for the MCAT. So the question is, who's charged? Draw the structure of the dipeptide GLY-HIS. And then I added an element because I love torturing you guys. Added the element of what is this in one-letter abbreviations? What is the charge on the peptide at pH 5.5, at pH 7.5? So these are questions we're going to be answering in this video. And I broke down what we need to know for this question. First, we have to know the amino acids. So we have to know both the 3-letter abbreviations and then the 1-letter abbreviations on top of the structures of the amino acids. So for the MCAT, you have to know the structures of the 20 amino acids, the 3-letter abbreviations, and also their 1-letter abbreviation. Pretty annoying, but you have to know them like the back of your hand. So this is a good way to kind of ease into it and get used to it. We're also going to talk about how amino acids are joined to form a polypeptide. A polypeptide is just a chain of amino acids. So how does that happen? How do 2 amino acids come together basically to bond to each other? And we're also going to be talking about how we can use pH and pKa to tell the charge of a charged functional group. So basically, if we know the pKa of like a functional group and then we know the pH, how do we find what charge it is? So first and foremost, we're going to talk about the amino acids. So we're going to talk about both the 3-letter and the 1-letter abbreviations of them. First, I'm going to start with the amino acids that are electrically charged. So here we have the positively charged ones. We have arginine, we have histidine, and we have lysine. And obviously we have the 3-letter abbreviation here, here, and here, and then we have the 1-letters R, H, and K. 2 things I want to point out from this section is that arginine has a guanidinium group here, and these groups are just differently named. They're just special groups for the side chains for certain amino acids that you have to know for the MCAT. They just like, just in case they throw a multiple choice question on there, they're like, all right, what group is this? And I think I've seen it on practice material, but they might throw like, oh, what group is this? They might have this show up or like show you the amino acid arginine, and then you have to say like, okay, A, guanidinium, that's what it is. Another one is histidine has an imidazole group, so it's this R group. Some negatively charged amino acids, we have aspartic acid and glutamic acid. These are interesting because they always tripped me up when I was in biochemistry, because aspartic acid is D and glutamic acid is E. It's not really, doesn't make sense to me. Doesn't come easy, such as like with histidine, it's H, so that's just easy. It's just histidine for H. For serine, for example, is S. It's the first letter. For these two, it's not the case, so I would have to commit these to memory. These took the most work for me when I was studying for the MCAT to commit to memory. And then we also have the amino acids with polar uncharged side chains. We have serine, threonine, asparagine, and glutamine. Now these are the hydrophilic or water-loving side chains on top of the ones that are polar or that are charged up here that we already talked about. And they're all going to be water-loving. They're all going to be at the surface of proteins touching the aqueous environment outside that has water in it. We also have cysteine, glycine, and proline. Now this is under special cases because these behave a little differently. Cysteine has a thiol group here, the SH, and that can form disulfide bonds. Holds quaternary structure of proteins together, holds subunits together for proteins. Glycine is an achiral amino acid. It's the only achiral amino acid. It has an H as a side chain. And proline has this connection to the amino group, the alpha amino group, that none of the other amino acids have. it kind of like loops around and forms this ring. So then we can talk about the amino acids with hydrophobic side chains. So these are the non-polar side chains. These are the water-hating side chains. They love going in the middle of the protein. They clump in the middle and they're away from the water. And there's an effect that I want to talk about in another video. It's called the hydrophobic effect. It allows proteins to form into their three-dimensional structure. And these are the amino acids that are responsible for the hydrophobic effect. It's all the hydrophobic amino acids coming together and they just want to clump up in the middle and they want to be away from all the water. So these are the ones: alanine, valine, isoleucine, leucine, methionine, phenylalanine, tyrosine, and tryptophan. A special group that I want to mention is in tryptophan is the indole group. It's this like double fused group with an NH. Again, they might ask it, what group is this, or what you know. You might have to classify it, and just in case you want to know it and commit it to memory, maybe just make a flashcard on these groups by themselves, like, oh, what group is this, what group is this, what amino acid is it from? This seems daunting, but we're going to mention amino acids over and over again throughout these videos, and you're going to get used to it. There's a lot of commonalities between them. Something else I want to mention is how is a peptide bond formed. So amino acids like this and this, so these are just two glycines right here. Amino acids are bonded through peptide bonds. So this is the bond right here. It's a covalent bond, it's a peptide bond, and it happens between these two. So I want to just quickly go over the different parts of an amino acid so this gets a little bit easier to understand. So we have the alpha carbon right here. It's called the alpha carbon. This is the alpha carboxy group, and this is the alpha amino group. So we have the carboxyl group, we have the amino group. Then we have a hydrogen, which all amino acids have. So this, and this, all amino acids have. The carboxyl, the amino, and the hydrogen. But the side chain is what varies from amino acid to amino acid. It's different for each amino acid. But how this works is that amino acids are built from the N terminus to the C terminus. What I mean by that is N terminus means from the amino group to the C terminus to the carboxy group. So here we have this carboxyl group, we have this amino group, and what you have is a loss of H2O. You have a loss of water. So this is a dehydration reaction. You have a loss of water, and it forms this peptide bond. So this is how you join different amino acids, how you put them together into a polypeptide chain, a chain of amino acids. So this is just a picture to show what I talked about. So this is the N-terminus. This is how a polypeptide basically forms. We have N-terminus right here. We have C-terminus on this end. It goes from N to C. Here we have glycine, a peptide bond right here. We have a peptide bond right here. And then we have valine and serine. So these are just different amino acids. This is how they're joined together. This is a tripeptide right here. If we had 2 amino acids, dipeptide, 3 amino acids, tripeptide, and so on. So something else I want to talk about is what's pH and what's pKa and how do they relate to each other. So from general chemistry, something I love about biochemistry is that it's a combination of gen chem, of organic chemistry, and biology. It's just everything all at once. So if you don't know what pH and pKa are, I can link some videos in the description, but just very quickly, pH is the negative log of hydronium ions or hydrogen ions. Now this is just, this tells you the acidity of a solution. pKa is the negative log of the Ka, and the Ka is the acid dissociation constant, which basically tells you how strong an acid is. So if the Ka is really high, that means a lot of the acid dissociates into the products, right? Because it's products over reactants. So if you have a lot of products, then the Ka is going to be really high in this fraction. So that's just the basic gen chem definitions of it. But what do they have to do with each other? Well, it turns out that pH and pKa are related when you're talking about functional groups. So like we talked about the amino group of an amino acid, the carboxylic acid group, the hydroxyl group, and we see that when the pH is less than the pKa, so each of these groups has its own pKa. For the amino group, our pKa is going to be a 9.5. For carboxylic acid group, our pKa is going to be a 2. And hydroxyl group, I'm not 100% sure, but it gets the point across anyways. So if the pH is less than the pKa, so say we know the pKa of amino group is 9.5, so say the pH is 2. So obviously the pH is 2 and the pKa is 9.5, the pH is less than the pKa, this is going to be protonated. It would be the other way around. If the pH was greater than the pKa, it would be deprotonated. So this is really, really important when we're talking about it, in that this graph kind of lets you, this seems like a busy slide, but let me break it down for you. This slide exemplifies what the relation between pH and pKa is. So as I mentioned, the pKa of carboxylic acid is a 2. The pKa of amino group is a 9.5. And the way this works is that this pKa1 is of the carboxylic acid right here, and this pKa2 on this graph is the pKa of the amino group, which is 9.5. Now these are relative values, so if you read this graph, this looks like a little bit more than 2, this looks like maybe not 9.5, but these are just approximate values, so just bear with me. When our pH is less than our pK1, we know that our carboxylic acid group is going to be protonated, right? We already know that the pH is less than our pK2, which is our amino group, so our amino group is already protonated. So both groups are protonated, and that's the amino acid we have right here, right? And we're not considering the side chain, which is the R group right there. We're not considering that, whatever that might be. We're just talking about the carboxylic acid group and the amino group. And then here, we become our Zwitter ion form. So a Zwitter ion form is basically a dipolar ion that amino acids can become, where the amino group is positively charged and the carboxylic acid is negatively charged. And the reason this happens is that the pH is becoming more than the pK1, which means the pH is going above 2. The pH is going above the pKa1 of the carboxylic acid. So that means it's going to become deprotonated. And the amino group is still going to be protonated because we're still below, the pH is still below the pK2, which is the pKa of the amino group. So now we're going to have a Zwitter ion, we're going to have a dipolar ion, we're going to have a negative, and we're going to have a positive. Here, once we cross pK2, now our pH is above both pKa's, so then both are going to be deprotonated, and we're going to have fully deprotonated. So a way I like thinking about this, at least how I thought of it in my biochemistry class, was that when pH is less than pKa, you have an abundance of these H+ ions, these hydrogen ions. So as I talked about, pH tells you acidity, and acidity comes from hydrogen ions, right? A lot of hydrogen ions coming around that makes it more acidic. So when pH is less than pKa, you have so many hydrogen ions are in solution, like way more hydrogen ions, than pKa. Look, a lot, when pH is less than pKa, you have so many hydrogen ions. And this carboxylic acid group and this amine group can go and they can just, take a hydrogen ion. There's so many of them around, they can just take it and they can protonate themselves. So here, this is what would happen, right? In both cases, you protonate yourself. You add an H+. When a pH is greater than pKa, well, you don't have that many hydrogen ions around, because remember, the lower the pH, the more acidic it is, right? The higher the pH, the less acidic it is. So our pH is so high, we don't have that much acidity around, we don't have that much H+ around, and our groups can't grab onto anything. There's not enough. So they become deprotonated. They're like, okay, fine, whatever. There's not enough H+ for us to grab onto. We're just gonna let it go. So this is the sad face, this is the happy face. Obviously, either way you want to think of it, that's how I like thinking of it. It makes more intuitive sense to me. But this concept is useful when we talked about amino acids with electrically charged side chains. So, obviously glycine, arginine, histidine, aspartic acid, glutamic acid. So the first three are going to be our positively charged amino acids. Our next two are going to be our negatively charged amino acids. The thing is that for biochemistry, at least in my class, what you want to know is the pKa of the side chain of histidine. You don't really have to worry about the rest of these. I haven't seen them ask like a question or something, information that they haven't given you about the pKa's of these other ones. They'll give you the information, but histidine is a pKa you want to memorize. Histidine side chain has a pKa of 6. So keep that in mind, note that down somewhere. So now we've learned basically everything we need to know to answer the question. So again, the question is draw the structure of the dipeptide GLY-HIS. So now that we know the 3-letter abbreviations, we know that means glycine and histidine. Then I ask, what is the 1-letter abbreviation? What is the charge on the peptide at pH 5.5? And what about pH 7.5, right? So this is our workspace. Here we can draw the structures of both amino acids. So here we have glycine. Here's the amino group. Here's the carboxylic acid group. Here's the hydrogen. And this is the side chain. Again, glycine is the achiral, most simple amino acid. So it will be glycine, GLY-G. Here we have histidine that has our imidazole group, as we spoke about. And this imidazole group is ionizable, meaning it can become positively charged. And here we have the amino group, the carboxylic acid group, and then we have the imidazole group, and then histidine is histidine, HIS, and H. So obviously G is going to be our 1-letter abbreviation for glycine, and H is going to be a 1-letter abbreviation for histidine. Now if we were to put this in a dipeptide, if we were to put this together, we form a peptide bond, and it looks like I missed a D at the end. One second, let me fix that. Form a peptide. bond. Ugh. There we go. We form a peptide bond in the middle here, and this would be the peptide bond, the C double bond O bonded to the NH. This would be our peptide bond. We would go from N terminus to C terminus. Again, amino acids are built from N terminus to C terminus. And we would have the amino group. This would be the glycine right here. And then this carboxylic acid group would attach the amino group of the histidine. And then it would be the side chain of the histidine with the C terminus, the carboxylic acid ending with the C terminus. So now when we talk about the pKa's of each of these, the pKa's that we really have to worry about is the imidazole group right here, which has a pKa of 6. We have to worry about the carboxylic acid group right here, which has a pKa of 2. We have to worry about the amino group right here, which has a pKa of 9.5. So these are the numbers that we have to keep in mind when they mention pHs. How do we solve it? We already know. When pH is less than pKa, we know that it's going to be protonated. When pH is greater than pKa, we know it's going to be deprotonated. So that's the same concept that we're going to have to keep in mind. So here at a pH of 5.5, we know that the pKa of the amino group would have to be, the pKa of the amino group is 9.5. So right here, pH is less than the pKa of the amino group. So this would have to be positively charged. We know that the histidine imidazole group has a pKa of 6. So again, the pH is 0.5 less than the pKa, so it's going to be positively charged. And then the carboxylic acid has a pKa of 2. So the pH is higher than the pKa, we're going to have a deprotonation. So here, we have a positive charge, and a negative charge. 1 plus 1 minus 1 equals plus 1. So this would be a plus 1 dipeptide at a pH of 5.5. Now, what would be the case at a pH of 7.5? So again, amino group has a pKa of 9.5. So pH is less than the pKa, so it's going to be protonated. So it's going to be a positive charge. The imidazole group of the histidine has a pKa of 6. So now the pH is actually greater than the pKa. So now we're going to have a deprotonation, but we're not going to have a negative charge because when the imidazole group is ionized, it's only positively charged. When you take a hydrogen away, when you deprotonate it, it just has no charge. It doesn't have a negative charge, it just has no charge whatsoever. So this has no charge, this has a positive charge, and the carboxylic acid group has a pKa of 2. So then the pH is way greater than the pKa. So now we're going to have a deprotonation, so negative charge 1 minus 1 is 0. And we've answered all the questions. Now we know how pH and pKa relate to each other. We know the amino acids. We know the amino acids. We have to know their classifications. We know that how peptide bonds are formed. We learned all of that. And I hope you enjoyed this video. I hope you found it interesting and you liked the form of answering the question. Let me know what else you would like to see in the comments. I'll keep these up. I'll keep these, I'll read the textbook and I'll keep kind of seeing what the most important concepts are and make these question videos from the textbook. I think it's really informative and it's kind of keeps you engaged. So yeah, let me know what suggestions you have, what I should add, what I should remove, and I'll read all your comments. Please make sure to press like on the video, subscribe if you want to see more of these types of videos, and thank you for watching. Bye.